Okay, to all of you, let's discuss chapter number 8. Let's go to the slideshow. So in this chapter, we are going to talk about some of the basic terminology which is used in the topics when we talk about the metabolism. So we'll be talking about how the energy is extracted wheat. So that all basically involves the catabolism. Um, so okay, very first we need to know is what is free energy change. So in like if suppose this reaction A plus B, these are the reactants and they get converted into the products like C and D. So in this kind of a reaction, if we want to calculate the free energy change, how to do that? So we calculate the free energy change for this reaction. And first of all, let's understand what these are. So delta G naught means the free energy change. The free energy change under standard conditions, and C and D, these are the concentration of products, and A and B, these are the concentration of things. Delta G naught is energy change under standard conditions. Now, first of all, what is the free energy change? What does that mean, and why are we interested in knowing the free energy change? So, free energy change in a reaction is the change in the energy, is the change in the energy when the reactants get converted into products. So, basically, it's created subtracting the free energy by subtracting the energy of the reactants from the free energy of the products. So, if you subtract the free energy of reactants from the free energy of the products, you get energy change in a reaction. So, how does it helps you? So delta, and it is the free energy change is designated as delta G, the change in the free energy. So if the free energy spontaneous, if the delta G change is positive, there is non-spontaneous reactions. So we will start spontaneous and non-spontaneous reactions. So if we want to calculate delta free energy reaction, we can do by this equation. This is delta G under the standard conditions. And what are the conditions? Standard conditions are the conditions of pressure, which is 1 atmosphere, and the solute concentration is usually 1 molar. So these are the standard conditions, and then we can calculate delta G free energy change. And in this equation, R is the constant and T is the absolute temperature, and this is the log value. So if you know all these values, you can put and you can calculate delta G. So as I said, if delta G is negative, those reactions are spontaneous reactions. Spontaneous reactions are which do not need the input of energy, and those reactions take place on their own, and those reactions actually release energy. So they are also called exergonic reactions. And on the other hand, there are reactions which are non-spontaneous, which means those reactions need the input of energy, and those reactions are called endergonic reactions because energy has to be given for that reaction to take kind of reaction called non-spontaneous reactions, and the delta G is positive for those reactions. And some reactions which are endergonic reactions, which means those reactions need of energy, and reactions are exergonic reactions. So there is a coupling of reactions takes place inside our cells. Endergonic reactions which need input of energy, they are coupled with the reactions which release energy. So in our cells, endergonic reactions, they are coupled with exergonic reactions. Here example, in our cells, glucose gets converted into glucose 6-phosphate. So that is an endergonic reaction. That reaction needs the input of energy, and that reaction can be coupled with hydrolysis of ATP. So ATP can get converted into ADP, and this reaction is ATP hydrolysis, and this reaction is an exergonic, and this reaction releases energy. So these two reactions, this endergonic reaction is coupled with this exergonic reaction, and this is the way reactions can take place inside our cells. The metabolism, all reactions which are going on in cells, they basically constitute our metabolism, and the two reactions going on. They are catabolic reactions and the anabolic reaction. Catabolic reaction means when some of the molecules convert in some of the molecules, some bigger molecules, they convert smaller. And anabolic reaction, like those smaller subunits or the smaller monomers, they actually are linked together, and they bond together, and they form some big macromolecules. Those reactions they are called anabolic reactions. So catabolic reactions, they are exergonic reactions, and anabolic reactions are reactions in which energy is required. They are the endergonic reactions. So this explains catabolism. So fats, polysaccharides, proteins, all these things which we eat in our food. In this, so these things we eat in our food, and they first of all get broken down into their simpler monomers, like peptides get converted into sugars, proteins get converted into amino acids, and glycerol. So a process was oxidation. Talk about what oxidation is, and these kind of processes of energy process. Anabolic action, these small units, polymers like into proteins, can get converted into polymers, and action involve action, and these are energy requiring processes. Now, what is oxidation? Oxidation is loss of electrons, reduction is gain of electrons. So, what is a reducing agent? It is that substance that uses that substance which gains electrons. Now, here is an example of oxidation and reduction. So, here is zinc. So, when a zinc atom loses electrons and it gets converted into ion, the zinc atom loses and gets converted into zinc ion. This is called oxidation. And copper ion, when it accepts electrons and gets converted into copper atom, this is called reduction. Now I'm going to talk about two coenzymes which we have already talked about in the chapter on the enzymes. There are coenzymes NAD plus and NAD. These are the coenzymes we called in our metabolic, in our basically the cell process, and enzymes which have vitamin E, and they have electron receptors. So in the food we eat, it undergoes process of cellular respiration, and in this process energy is released. Energy is released from the food we have eaten, and the energy in our food is in the form of high energy electrons, and these electrons, they're carried by NAD plus and FAD. These electrons, these high energy electrons which are present in our food, they are actually carried by NAD plus and FAD. These are the two coenzymes, and they are being taken by these electron carriers to the inner membrane of mitochondria. So this is basically shown Na plus dinucleotide. So you can see here that it is here it is accepting one hydrogen atom, and the hydrogen atom has one electron and one proton, and NAD plus gets converted into NADH. And similarly, this is FAD. This is another coenzyme. So you can see it can accept two hydrogens, and one can attach here and another can attach here. Hydrogen atom has one electron and one proton. So basically, hydrogen atom is basically is a representative of the electron, because every hydrogen atom has one electron and one proton. So FAD gets converted into FADH2. And this is, yeah. So now we're going to talk about ATP. So what is ATP? ATP is the currency. So as I said, the energy is in the form of the high energy electrons in our food, and this energy is in the process of cell respiration is converted into ATP. ATP is adenosine triphosphate. I can show you its structure. This is the structure of ATP. This is the ribose sugar, and here are attached the phosphate groups. So why this is energy currency and how does it carries energy? Because this compound or this chemical has three phosphate groups, 1, 2, 3, and all these phosphate match on them. So to make a bond between these phosphate groups, because they're all negatively charged, they're going to repel each other, a lot of energy is required. So the bonds which are formed between these negatively charged phosphate groups, they're called phosphoanhydride bonds. So a lot of energy is required to make these bonds. So the energy which is of that was ADP into ATP. So this is, so in us we have adenosine diphosphate. So the food we have eaten, the energy which is released, that energy, because this process, this catabolic process is exergonic, that energy is used to take bond with phosphate group and between this phosphate group and AD, and that results in the formation of ATP, adenosine triphosphate. And this ATP, which is the energy currency, so this is a lot of energy in it. That energy is used for the anabolic processes, and these anabolic processes, they are endergonic process. They need energy. And in these processes, for these processes, the energy comes from ATP. ATP gets converted into ATP. So here, this is a structure of ATP. So this bond is going to be broken, and this phosphate is going to be separated, and this compound will be converted into ATP and will be released, and then will be. And this basically shows overall catabolism and anabolism which is going on in our cells. So the food we eat, first of all, so the food we eat, eat food for getting energy, and that food goes through a process of cellular respiration. That cellular respiration has different stages. Glycolysis is number one, followed by that citric acid cycle, then is the oxidative phosphorylation. So in all steps, energy is released ADP, and ATP are produced here as well as used here. As well as in all these steps, NAD plus, which is the electron carrier, it also gets reduced and it forms NADH, and it basically carries NAD plus gets reduced to NADH and it carries the electrons, and these high energy electrons, they come from the food. They come from the food we have eaten. Carried here, some of the electrons are carried here. So in the glycolysis, the electron carrier involved is NAD plus. In the citric acid cycle, the electron carrier involved is NAD plus and FAD, and all these electron carriers, they carry the electrons to the inner membrane of mitochondria, and this process of oxyphosphorylation takes place and a lot of ATP is produced. On the other hand, anabolic reactions, they need energy. So in these reactions, ATP is converted into ADP, and NADH is basically oxidized back to NAD plus. So the chapter, basically it involves some terminology. That's all for this chapter. If you have any question, you know.